right, so I'm going to talk about bacterial and seed health issues. This is kind of a long-winded PowerPoint, so if you need to get up and get something to eat or drink or use a restroom, please feel free. Oh, does this not?
It doesn't, I had to use the keyboard. That was very
Oh,
strange. Yeah.
Okay, well, okay. All right, so a couple of introductory slides. We're specifically going to look at bacteria and parasites in here for this PowerPoint presentation and correlate that with water quality to some extent. In hatcheries, bacteria is our primary concern. And as you move to nurseries and grow out, parasites become more of a concern. Not that bacteria aren't a concern, but there's really not a lot you could do about it at that point. Water quality can have an impact on both of these. So temperature changes can affect your bacterial population, and salinity can affect your parasitic population. In addition, your water quality parameters like high ammonia levels or low dissolved oxygen levels are stressors, and those stressors can impact an organism's ability to fight off an infection. So if there is too much of a bacterial parasitic problem, the animals will have a hard time dealing with it. One thing to keep in mind is bacteria are always present. Most of them are helpful or necessary. Think back to the water quality presentation. You've got your nitromonas and your nitrobacter that convert ammonia to nitrites and then nitrites to nitrates. So that would be an example. Probiotics are another examples. But we do have some bacteria that we call opportunistic. When they get to high enough levels, they can cause diseases and a problem in aquaculture. And whether it's shellfish or some other type of aquaculture, in a marine aquaculture operation, Vibrio is the culprit and the one that we really pay attention to. We do this by using a media called TCBS. It's a Vibrio specific media. It's a green media, as you can see here with the plate, and it's beneficial because it allows us to distinguish between pathogenic Vibrios and non-pathogenic Vibrios, with the pathogens appearing green and the non-pathogens appearing as yellow colonies. That's quite helpful. We are recommending, and you'll have some literature to go over that, monitoring hot spots, things that really can affect everything, and we'll go over that a little bit more later. For instance, things like your algae cultures and your source water should be free of Vibrio. So what type of monitoring did we do? In year one, we did quarterly sample. You saw that chart right up the very first PowerPoint. And we monitored both bacteria and seed health on a quarterly basis. In year two, we kicked the bacterial sampling up to a monthly basis. We already went over how we monitored bacterial samples, and we took a look at multiple things: water, clams, algae. For seed health monitoring, we used a process called histology, which makes takes live animals and makes a slide of them. You may remember this from high school biology class. The benefit of that is you can look at multiple issues at a time. So we can see bacteria, but we can also see parasites. We can see if the animals are feeding. We can see the condition of the digestive tract. So it gives us a good overall picture of everything. So the number of samples that we collected in year one and year two vary somewhat because, as I said, we went to monthly sampling with the bacteria in year two. And you can see how we've kind of broken it down by year, by hatchery and nursery. And the first year, we took quite a few nursery samples. And the second year, we didn't have a lot of bacterial samples for the nursery. Water samples could have been anything. That can be your tank water, larval tank water, broodstock tank water, source tank water. I'm not distinguishing that here, but we definitely had more water samples than we did clams or algae samples. For whatever reason, in the second year, not as many people submitted algae samples. So let's take a look. You're going to see a lot of tables in this rest of this presentation. So just breaking it down by year one and year two. When we talk about percents here, I'm talking about the percent of hatcheries, nurseries, the percent of facilities where we saw this. So this isn't an increase at one particular hatchery or nursery, but this is the percent of facilities. So for instance, in year one, 70% of the facilities at some point in time during the year reported or we saw high Vibrio counts. And larval tanks, 60% of the facilities, 100% of the brood tank samples that were submitted had high Vibrio counts, and so on and so forth. Another thing to mention is when we look at our source water or reservoir water, that typically we had higher Vibrio counts in surface water than we did in well water, which is to be expected. You are supposed to have less parasites, less bacteria and things like that in well water. So we're going to take a look at seasonal changes in year one and year two in both hatcheries and in nurseries. So in year one, again, this is an increase. Let's start a disclaimer here. We looked quarterly, so we've got winter, spring, and summer. So low Vibrio concentrations were seen in samples collected in the winter pretty much across the board. And there are reasons for this, and we'll talk about that. But as you go from winter into spring, you see that 60% of the facilities, for instance, saw an increase in bacterial populations in their larvae. Now, this is the animals themselves, and 60% in the corresponding tank water, and so on and so forth. Where you see a decrease, that simply would indicate that there were some issues in the winter. And so that's why you're seeing a decrease in the spring. So there would have been a facility or two that reported that had higher levels of Vibrio in their brood tanks in the winter. As we go into spring to summer, I was kind of surprised by this as I started crunching the data. I would have expected to continue to increase because of the temperature. It didn't. In fact, it either remained steady or we saw a decrease almost across the board, either no change or a decrease. So now we're going to look at year two, and in purple, for comparison's sake, is year one. So we're going to have them side by side here. Again, low Vibrio concentrations seen in the winter. So you can see a trend. There are some differences. I don't know that these would even be statistically significant because we only had 10 facilities that we were looking at. But you can see the pattern is the same, where you have this increase from winter to spring, a few, either it stays similar or decrease. And then again, as you go into spring into summer, you don't see a big increase. In fact, you see a decrease. In year two, it seemed to be even greater than in year one. And I'm going to move to nurseries. Now, nurseries, remember, we only had enough samples collected in year one to really make any judgments. So we're only going to look at year one. In the purple, now I'm comparing hatcheries to nurseries. We had some facilities that were just nurseries, just hatcheries, and we had some that had both. So we're not looking at algae or brood tanks at this point. So it's really only the animals themselves or the wellers that they're sitting in. Again, you see this low concentration in the winter, and you see an increase from winter to spring. Contrast it with the hatcheries. For the animals, it was very similar. Go spring to summer for the animals, very similar again, between hatcheries and nurseries. We did see this increase in Vibrio populations in the wellers, and I think that just reflects what's going on in the water, because we are talking surface water. You don't have any control about the Vibrio population in the Indian River Lagoon or Tampa Bay or wherever you're located at. So going back to some of these water quality parameters that you had seen in earlier slides, increased bacterial growth occurs as temperatures rise. Bacteria multiply much faster the warmer it gets. You know, that's why a lot of times hatcheries shut down at the heat of the summer. one of the reasons that they do. So what can you see in the summer? You can see a rise in your temperature, a rise in your salinity, a decrease in your dissolved oxygen levels because cold water holds more oxygen, as was pointed out earlier. So I'm going to show an example here of what you can see. So here we are in February. You've got nothing in your animals. This is pretty typical, very little in the tank water. And all of a sudden, here it's spring, you see a rise in your bacterial population in both larvae, or both your post set and your wellers. You don't really see, as I said before, a change from spring to summer, but in this particular case, and in some of, not all facilities, but in some facilities, we did see an increase in the number of pathogenic Vibrios. So it wasn't an increase or decrease much or change in Vibrios per se, total Vibrios, but the pathogenic ones did increase in this particular facility. We didn't see that everywhere. We saw it in a couple of facilities. So now I'm going to move on to the seed health pathology reports, which if you were a participant, you received this. And again, these were done quarterly in both year one and year two. Again, to point out the dates here. Winter. Great. Not only do we not have bacteria, but we don't have any other issues going on, right? June. We do see an increase. Now, these aren't, earth shattering. We're not going from 0 to 100, but you do see an increase in condition of the animal. And you do see an increase in bacterial prevalence within the tissues of the animals. And I make some comments about, they're feeding, they're not, we've got other things going on. I'm going to walk you through a little bit what we would see with histology. Some of you may find this boring. I kind of like it. I find it fascinating. I do a lot of it. So this is a baby clam underneath a microscope, a whole clam. You see your abductor muscles here. You've got your kidneys and your heart back here being protected, gills, digestive tract over here. This is a close-up of the digestive tract. And on those reports, we talk a lot about the digestive diverticulum. This is where all the nutrients are delivered throughout the animal's body. This is a healthy one. When we talk about tissue disintegration, this is what we're talking about. So this is a tubule, it's blown up, a higher magnification, and you can see how it's not intact, it's breaking down. You can see another one breaking down over here. You can see gaps in between the epithelial layer, and it's sloughing off into the interior of the lumen. And you can also see little purple dots, which are bacteria. Okay, next to it, you do see some healthy ones. But what this means is this is non-functional. So this particular digestive tubule isn't working. So if you start to get a lot of these, a few of them's okay, but if you start to see, 25, 50% of that, then your animal's not going to get the nutrients it needs, and that there's going to be an issue. Yeah.
Would it be able to grow into more cancerous tissue?
You mean would it be able to replace the tissue?
Yes.
Yes, they can recover from this if that's what you're asking, if it's not too advanced, not too crazy.
Right.
So also talking about condition, we also mentioned the digestive tract. Yes?
Sorry, on the previous slide, were the clams contributing any value that, or was that just...
Well, all these were taken from the project. I would have to go back and look and see who I took this picture from, when and where, and all that stuff. Good question. So here's another example of conditions. We've got the digestive tract itself. That's an intact along the side of it with the digestive tract inside. That's the lining. This would be an example when we talk about hemocyte accumulation of a swollen digestive tract, lots of hemocytes here, the purple dots, which means there's something going on there, probably bacteria, since I don't see anything else jumping out at me. This is another thing that we talk about in the reports. It's called concretions. This is an interesting one that I could spend a lot of time on it, but I'm not going to because this is a long enough PowerPoint. But this is actually a pathological condition. It's not caused by a pathogen. They really don't know what causes it. It's usually environment, like toxins and things like that. And toxins can also break down the diverticulum and digestive tract as well. This is an example of a cluster of hemocytes again, attacking bacteria and getting ready to form a granuloma. And here are pictures of filamentous bacteria that we saw in one sample. Granulomas being formed. We talk about granulomas and hemocytes around it. And here is a worm, metazoans or worm parasite that's being sequestered. So the animal tries to wall these things off in its tissue as they occur. And it can do that as long as they don't get too overwhelmed. So that brings us to hatchery seed health. Yes, Richard.
Yeah. What sites not animals?
This, usually you see the granulomas probably more in post set. It's as they get older that you see that advance, but again, I'd have to go back and double check on that. So if we take a look at, that was just all an introduction to, so that you would have a visual about what I meant when I talked about those things or on your individual reports. So winter to spring, again, just to point out that very similar to the Vibrio issue, winter, 70% of the hatcheries were not reporting any issues in the winter. And then we see this increase in condition variation from winter to spring. We also see an increase in some of the pathogens. This is reflected in the Vibrio reports as well. Going from spring to summer, again, you can see the difference in the spring. Only 20% of the facilities had no issues. So they would have had one of those nice clean reports. In the summer, you see 40% that had no issues. So again, this is going that spring to summer where you're not seeing an increase. In fact, you're starting to see a decrease. Except perhaps in some of the pathogens. But as far as a condition, now these things can go hand in hand. Bacteria, hemocytes will go to fight bacteria. Together, they may form a granuloma. So some of these things kind of all go together. And I'm also not talking about the degree here. It's just these were seen. So some of them could have been very mild. Some of them could have been moderate. Some of them could have been heavy. Just to point that out as well. In year 2, again, we're going to compare year 1 and 2 using purple next to it. You did see a little bit of a different picture, winter to spring, but you saw a very similar picture between the 2 years from spring into summer. And you saw similarities when it came to pathogens. Again, just for comparison's sake, for whatever reason, in year 2, we had more facilities that had issues in the winter for whatever reason than we did in year 1. So let's move on to nurseries, because for histology and seed health, we did both hatcheries and nurseries in both years. Again, you can see this same trend in having most of the majority of nurseries, just like hatcheries, didn't have issues in the winter, but you started to see an increase in condition of the animals, negative condition as we entered into springtime. Very similar to the hatcheries, you saw an increase in the bacterial populations, very few decreases. And this is kind of interesting. You see a contrast here from spring into summer, where you continue to see an increase in issues in nurseries, where you don't in hatcheries, but the pathogens load is very much the same. Now we're comparing year 1 to year 2, and you can see that things were a little bit better in, well, I don't know if you could say better, it depends on what you're looking at, I suppose, from year 1 to year 2. You see less hemocytes, but again, you see patterns in most things where you get that increase winter to spring, and it either stays the same or you get a decrease from spring to summer. There are some differences between the years. So recommendations for hatcheries. Where to test for Vibrio. This chart is in the handouts that you get. It's also on the website. We've presented this at other workshops in the past. Where do you want to test for Vibrio? Well, your water and your algae are going to be fed to all the different systems, all the different sized animals. So if you're limited in your time and what you want to test, those are the two areas you should hone in first, because they're going to affect everything else. If you're feeding food that contains Vibrio, you're feeding that straight to your animals, you're going to expect to see bacteria in the digestive tract. Same with the water. If you're putting them in water where you've got an issue already, you're going to expect to see a problem. When to test? Well, obviously spring is a very good time to test, if you're going to do your testing, because that's when you really start to see the problems. Winter, you could get by without really looking at things because you don't usually see a problem in the winter. So what's an acceptable level of Vibrio? Ranges that have been reported where there have been no issues in the seed are in this particular chart here. Again, you can see what's your acceptable range for your water and your algae. Nothing. Pretty much nothing. And certainly no pathogenic green colonies. Now, when it comes to your tank water, that can be a little bit more of a population. I'm conservative and I like to go on the lower end of things. So if I start to see something climbing it about to 5,000, I get worried. And to me, that's a red flag, even though you can go as high as 10,000. But I wouldn't recommend that. But again, you can see the contrast between total, yellow, and pathogenic. You want very low counts. In your animals, again, very low counts. Can be higher than your algae, but you're looking for low counts. Now, as part of this project, we did encourage facilities to send us samples when they started to experience mortalities, heavy mortalities. And in year one, we had that 5 emergency samples submitted either at the end of the winter or into spring. So point that out again as being a hot spot. And in year 2, we had 3 submitted, 2 in the spring, 1 later in the fall. So let's take a look at year 1. All the samples that were emergency samples did show high levels of Vibrio. For some of them, we did histological confirmation. Interestingly enough, the populations in the tank water tended to be substantially lower than the clams. So here's an example of one of those emergency submitted samples. You can clearly see Vibrio counts that are not within acceptable limits. In the larvae, they were almost 100,000. You're looking at 100 as acceptable. I also point out the green, the pathogenic, it was higher than the yellow. And although the tank water was within acceptable levels, it's predominantly green. So it's the pathogenic ones that are looking at it, we're looking at. And for this particular facility, we did run histology at the same time. You can see that 75% of the digestive tubules were compromised. And you can see that we found that 26%, I think it is, of the clams that we looked at, they showed bacteria histologically. For that particular facility, they then sent us some algae and some of their source water at the same time we received those samples. We didn't always get that. Sometimes we just got the tank water and the animals. But here we got the whole picture. And we were able to find out that the algae was clean. It wasn't an algae problem, but that their incoming water was the culprit. I think it had levels of like 8,000 CFUs per mil of Vibrio. Yes?
What are the What was the standardization for the sample collection from the algae? I assume it was algae that was about the animals, but was it noted what species? Was it already pre-mixed?
So we did know what species it was. It typically was what they were getting ready to feed out. It was not their stock cultures. It was not the test tube cultures. It was what they were getting ready to feed. Yes, good question. Now, it's not always Vibrio. So in year 2, we had 3 emergency samples submitted. In 2 out of the 3 cases, there was no Vibrio in the tank water that was submitted or the animals. So there's something else going on. But at least this is, something that you can easily test for. And so I have no idea what was causing the mortalities in the other samples because we didn't find anything with other things that we went to test, any water quality issues, any pesticides or that type of stuff, but we didn't, necessarily test that at the same time as we had those emergency samples submitted to us either. So recommendations and kind of an overall summary. You can expect to see high Vibrio loads as the temperature increases. Parasitic loads are associated with high salinities. And as we noted before, low DO, high ammonia can cause additional stress. I took a look at the dissolved oxygen and the ammonia levels, and I didn't really see any correlation. All the correlations I saw were temperature and salinity. So one of the things to note, and I mentioned this previously, in the winter, why aren't we seeing issues? We've got wonderful conditions. Temperature's low, salinity's low, dissolved oxygen's high. We see the flip of that in the spring and going into summer. So if you're a hatchery operator, do what you can to lower your stress, particularly in the spring. So let's hone in on the spring. And I think one of the reasons why we may be seeing more issues in the spring than we really do in the summer is spring is very up and down. It can be hot in the day, cold at night. You might have 1 week where it's cold, 1 week where it's hot, and that alone is a stress on animals. So whatever you can do to keep a steady temperature, a consistent temperature. And as I mentioned before, that's the time to test for your hot spots. Be really cognizant about looking at your algae in your source water to make sure that's not a problem. For you nursery operators, I don't have a lot for you. Prayer, meditation, a cold beverage, that's all I got to offer you because you're kind of in Mother Nature's hand. It's kind of a pity, what's going on, but it's kind of hard to deal with it. So last slide, additional resources. Natalie will talk about this in a minute, but we do have bacterial centric handouts, including how much supplies cost if you want to make your own media, where you can get that from. You can get pre-made media. We also put together a video that's on the website that tells you how to sample, how to make media, how to plate your samples, and an interpretation guide. So that is all on there as well. And I've had a lot of questions in between, but 1 or 2 more.
So in measuring the increase and decrease in the concentrations of the Vibrio, between your years, I assume when these samples were taken and the values were recorded, you reported these back to the aquaculture facilities. And did they take any steps to decrease the amount of Vibrio?
Well, now that's, if like in emergency samples, they definitely got feedback. If I saw high numbers, they got feedback. We didn't necessarily give much feedback right at the time if you didn't have an issue. So it was more if you had an issue, we communicated with you. Whether or not and what steps they took, I don't know. Like I said, some of this surprised me and I really didn't get a good handle on it. So I really started looking at everybody's data combined. Those trends in the spring and then no additional trend in the summer, which really surprised me. I wasn't expecting that. Nicole?
Did you look at grow out product at all as well?
No, we did not.
Well, Some of the date ranges seem very similar to when I'm having a giant mortality event in the past 2 years of my grow out and market product, the oysters. So
You know where I live and I know where you live, so. Maybe we can get together and do something about that. Okay.
One more? Good.
Do I have another one? Oh,
Is there anything known about the non-virulent strains of Vibrio, either, when they're not being actively virulent or if they don't have the capacity do they have an active role in the natural gut microbiome of the clams at all?
well, there have been some people I know who've done fish work years ago who actually would try to make a probiotic composition out of the non-pathogenic ones. That's a lot of
Very
work unless you have the facilities to do that. And some people don't like to see any Vibrio. They don't care what the color is. They just don't, they're done with it. All right, is it Shirley's next up? Okay.